Hello everyone, in this video we are going to study the structure of secondary oocyte. The secondary oocyte is also called as unfertilized ovum. So related with this unfertilized ovum or secondary oocyte, when the Graafian follicle get formed, the fully grown Graafian follicle get formed, at that time the secondary oocyte get formed. Okay, before the rupturing of the Graafian follicle, it gets the primary oocyte get converted into the secondary oocyte before the ovulation. Before the ovulation, just near about 5 hours before it get formed. Okay, the second it get when the LH surge occurs, during LH surge, the Graafian follicle ruptured and the structure which comes out from the Graafian follicle and from the ovary is nothing but the secondary oocyte. So this is the secondary oocyte. The secondary oocyte is also called as unfertilized ovum. Some people are more commonly we are calling it as a ovum, but it is not a complete ovum because it is arrested at metaphase. It is not completing its cell division, so we named it as a unfertilized ovum or secondary oocyte. Clear? So it is the largest cell in the body of a human, and it is spherical and non-motile cell. Its diameter is about 100 to 200 micrometers in a diameter, or zero point 0.1 to zero point 0.2 mm in the diameter. It is related with this. So at its center site, it having a nucleus. This is the plasma lemma, or it is also called as a oolemma. It encloses the cytoplasm. This is the cytoplasm or ooplasm. Cytoplasm at its center site, cytoplasm or ooplasm. The human egg having abundant amount of cytoplasm and very little amount or least amount of yolk or free from yolk. So this egg type is called as a microlecythal egg type. So human egg is a microlecythal egg type, can be asked for a single sentence. Okay, so everyone will keep in mind to whom we call as a microlecythal egg, which having abundant cytoplasm and free from the yolk. Such a type of egg is called as a microlecythal egg type. Okay, so because of the abundant cytoplasm, the nucleus moves towards the periphery. That means here is nucleus is kept at the periphery, and this nucleus is arrested at metaphase. So this is the nucleus. We will name it as a nucleus, but it is arrested at metaphase. It is arrested at metaphase, right? Then this cytoplasm is of two parts, divided into two parts. At its periphery, near to the plasma membrane, it becomes granular. It becomes granular, and this granular cytoplasm is called as an exoplasm. It is called as an exoplasm, and which is at inner side is called as an endoplasm. Okay. Further on, this cytoplasm is covered by this layer is called as a plasma membrane. Plasma membrane. Or plasma lemma, or it is also called as a oolemma. It is also called as a oolemma, right? So then just around to it, there is a perivitelline space. This is a perivitelline space. Okay, oolemma, or it is also called as a vitelline membrane. One another name of a plasma membrane is a vitelline membrane. Okay, just around it there is a perivitelline space. This perivitelline space is filled with a fluid called as a perivitelline fluid. It is called as a, here will be a perivitelline fluid. Perivitelline fluid. In this perivitelline space there is a polar body. This is the polar body. When primary oocyte get converted into the secondary oocyte, at that time this polar body get formed into the Graafian follicle. Okay, then this vitelline membrane is covered by a non-cellular membrane which is secreted by this membrane, this layer, the glyco, and this layer is made up of a glycoproteins called as zona pellucida. This membrane is called as a zona pellucida. This zona pellucida layer is non-cellular. It is non-cellular transparent. But the unique feature of this zona pellucida layer, this zona pellucida, what is the function of this zona pellucida? This zona pellucida layer doesn't allow the wrong site implantation. It doesn't allow the wrong site implantation. That means it is not allowing the implantation in the fallopian tube. Okay, this is the function of this zona pellucida. As well as this zona pellucida layer having receptors which receives only the human sperm. This is very peculiar function because it is specificity is seen because of this receptors which are present in a zona pellucida layer. Okay, that means this zona pellucida only receives human sperm or just allow the human sperm to fuse the ovum or egg. Okay, then clear. And this zona pellucida is lysed by zona lysing enzyme which is present in the acrosomal cap of the sperm. Then this zona layer is surrounded by this layer of cells are called as corona radiata cells. Corona radiata cells. These cells are just formed from the cumulus oophorus. Cumulus oophorus is formed by the just follicular cells which get surround to the egg. Okay, when it get ruptured, along with the cumulus cells of cumulus oophorus, the secondary oocyte comes out. Okay, and when it comes out from the Graafian follicle, it converts into corona radiata cells. Clear? These corona radiata cells are stick with each other or glued with each other by hyaluronic acid, and this hyaluronic acid get dissolved by hyaluronidase enzyme, which is also present in a acrosomal cap. So this is the structure of human egg. Once again, we will revise. Human egg is the largest cell in the body of a human, and its diameter is about 100 to 200 micrometers. It having abundant amount of cytoplasm. It lacks centriole. Okay, here there is no centriole. As you know, the role of centriole is cell division, carry out the cell division. But this cell lacks the cell division, lacks the centriole. But during the cell division, but when it get fertilized during this process, along with the male pronuclei, the proximal centriole of a male, that means the sperm, enters in this, and then this undergoes division. Clear? So it lacks centriole, but it have an abundant amount of cytoplasm. It lacks yolk also. So the egg which contains maximum amount of cytoplasm and very little amount or least amount of or absence of yolk, that type of egg is called as a microlecythal egg type. So this cytoplasm is of two types. At its periphery, it is granular, it is called as the exoplasm, and its central side, which is clear one, it is called the endoplasm. And nucleus is centrally placed, and this nucleus is arrested at metaphase, and because of this only, it is called as unfertilized ovum or secondary oocyte. When it gets completing its further division, when nucleus will complete all its division, then we will call it as a ovum, right? Then this cytoplasm is covered by oolemma or plasma membrane or a vitelline membrane, right? Just around to it there is a perivitelline space which contains perivitelline fluid, and in just the perivitelline space there is a polar body. This polar body is formed in a Graafian follicle, right? Then the next one here is the vitelline membrane is surrounded by non-cellular transparent membrane called as the zona pellucida, and this zona pellucida is a non-cellular structure, and basically it having receptors which just allow the human sperm to fuse the ova, and it doesn't allow wrong site implantation. Then the zona pellucida is surrounded by corona radiata cells. These corona radiata cells are glued with each other or stick with each other by hyaluronic acid, and it gets dissolved only by hyaluronidase enzyme, which is just secreted by, or which is just released by the uh, human acrosome during the fusion, right? So this is all related with the human egg, unfertilized egg or unfertilized ovum or a secondary oocyte. Thank you.